All right, thank you. So hello everyone, and thank you to the Genealogical Society of Ireland, particularly Michael Merrigan, for inviting me to speak today. So isotopes and ancestry, exploring how archaeochemistry can answer genealogical questions. So today we're going to explore how isotopes, ancient DNA, and genealogical research intersect, particularly how carbon, nitrogen, and strontium isotopes can further address questions about someone's life and how ancient DNA can be used for remains identification, thus revealing someone's lost identity. We'll explore examples of this with a historic case of identifying siblings from about 200 years ago, a modern forensic case where a murder victim was identified, and the identification of a 1916 Irish nationalist, Thomas Kent. I'll be discussing various applications that archaeochemistry has within genealogy, but I will also cover potential bias within the research and also how funding opportunities and personal research bias can restrict and manipulate this information. And we'll end with time for questions and discussion. I'm actually really nervous and excited to talk to you guys about this stuff. So a little bit about me. As he said earlier, I have a PhD from UCD, where my research focused on the diet and migration patterns of Hungarian populations, particularly from the Bronze and Iron Ages. This was combined with other researchers' projects from different institutions to look at populations that span from the Neolithic to the medieval period and beyond. This ranges from about the 7th millennia BC to 1300 CE, or spanning from about 700 to 9,000 years ago. I've also analyzed a cross-section of an Ohio Native American population called the Turpin Indians, dated from about 1000 to 1750 AD, who were suspected of being one of the first populations to contract tuberculosis from indirect exposure to colonizers. And the bulk of my research uses isotopic analysis to compare demographic groups and cultures as a broad spectrum. So my interests include a host of scientific archaeological approaches, but my main goal is to add to the narrative of people by enriching their story about who they were and their life's experiences. So firstly, what are isotopes? Isotopes are variants of an element that share the same number of protons, but have different neutrons, which results in having different mass numbers. This differential is what we use to determine certain levels within a sample, such as bone, and compare these to other samples. There are over 3,000 isotopes, with about 300 naturally occurring. There are naturally occurring isotopes, both stable and radioactive, and there are artificial isotopes that are all radioactive or unstable, both of which are used within archaeological research. Some examples: calcium, mercury, titanium, lead, chromium, magnesium, uranium, and beryllium are all examples of isotopes. And some interesting isotopes are uranium-235. That's the fuel that's used for nuclear power plants. Cobalt-60 is used in radiotherapy. Iodine-131 can diagnose and treat thyroid disorders. And strontium-82 decays into rubidium-82, which is used in heart imaging scans. So isotopes in archaeology. You are what you eat, which is my favorite thing to say in archaeology when it comes to isotopes. The body incorporates chemical signatures, or fingerprints, so to speak, through the diet. These fingerprints allow archaeologists to reconstruct diet, nutrition, including weaning and breastfeeding practices, human and animal mobility, environmental analysis, carbon dating, and the provenience of artifacts. The questions they can address are endless, especially combined with other research tools like DNA. When discussing isotopes, most people think of carbon dating, which is the measurement of decay of C14, a radioactive carbon isotope. However, other isotopes are used in archaeology that can provide a plethora of data about both humans and other animals and the climate. In archaeology, we have calcium, carbon, nitrogen, oxygen, and strontium are the most commonly used. But for this talk, I will focus on carbon, nitrogen, and strontium, and a little bit of oxygen. So firstly, for carbon isotopes are incorporated by plants via their photosynthetic pathway. which is how we are able to distinguish the main groups in our analysis, as the different pathways utilize different types of fractionation processes. The subsequent chemical reaction results in a 3 or 4 carbon acid within the plant's own cells. These different pathways are used by different broad categories of plants, C3, C4, and CAM. And the plants often used in early agricultural practices like maize, millet, and sorghum are in one of these categories, C4. The bimodal distribution of values for C3 and C4 plants help archaeologists by reconstructing possible dietary composition ratios, and to look at the increasing presence of certain plant group signatures indicative of different human-plant relationships throughout the millennia. These values are also incorporated into our own bones, and the results, depending on its range, may indicate either a terrestrial or freshwater-based diet, a heavy plant-based diet, or other category differences like age, sex, and status access. This may also indicate proportions of plant and protein in the diet by combining with nitrogen isotopes. Similarly to carbon, nitrogen isotopes are incorporated into the body via diet at incremental or trophic levels along the food chain. For plants, nitrogen is integrated from the soil and can reflect other environmental signatures. As plants acquire their nitrate or inorganic ammonium from the soil, their nitrogen isotope signature will reflect the ground values. These are then reflected in the consumer's isotopes. The use of stable nitrogen isotope values in diet analysis is predicated on the assumption that humans obtaining protein from plants will have nitrogen value similar to a local herbivore, whereas humans consuming protein from animals as well as plants will have nitrogen values increased by a trophic level compared to their solely herbivorous counterparts. This is interesting because that implies elevated nitrogen values in humans may indicate an increase in the consumption of meat or dairy products, suggesting that the quantity and quality of dietary protein intake has an impact on nitrogen enrichment. There's some nuance on this. I suggested modifications to this model include both the effects of manuring and the incorporation of increased cereal nitrogen values by 1 parcent per mil. When coupled with carbon, you can see distinct substance strategies, with the hunters having the highest nitrogen and carbon values, especially compared to the agriculturalists. For strontium, which is my favorite isotope, mainly finds its way into an organism through its diet, as strontium is introduced via calcium-rich foods. However, it is still a reflection of dietary resources. For example, if an adolescent human were to consume migratory species like deer or any other non-local food or water source, their TM ratios may show more variability compared to the local geology because their dietary input has more variation. Different strontium values can exist within one individual, which can indicate potential regions in which this individual moved to or from. This is due to its uptake within our bones, teeth, and hair. As our bodies incorporate isotopes, bone, teeth, hair, and tissue have different values due to how quickly that material type grows itself. Bone, for example, replaces itself slowly over the years and can reflect a span of time during their life. When teeth are being formed prior to eruption, they lock in strontium signatures of their diet at that time, and compared to younger teeth, may provide evidence of movement. Hair grows quickly, and as it is formed from the scalp, each millimeter contains information from dietary strontium and other isotopes like oxygen. So the further you are from the root, the deeper you are looking into their past. Although strontium isotopes are incorporated through the food chain, this process does not produce the same fractionation factor like the trophic levels as carbon and nitrogen. This is the critical difference when interpreting these isotopes. Because of this, they can be compared to the locally available strontium levels found in geological samples. These signature maps help archaeologists and forensic analysts alike in determining potential home ranges of an individual, whether human or animal. The difficulty in applying this type of analysis of modern day samples would be due to globalization and the potential increased exposure to different geological regions through imported consumables and travel. DNA is the material that carries genetic information. After extracting a sample, it goes through a chemical process in order to quantitate it, measuring the amount for optimum sampling. Afterwards, amplification copies specific regions, known as polymerase chain reaction, or PCR, This makes copies in the millions of these regions, in particular short tandem repeats, or STRs. These are the sequences of DNA that differs between individuals due to its repeating sequence in different lengths and are separated by size during electrophoresis. The result of this created a DNA profile made up of numbers corresponding to its genotype for each STR. These can then be compared to an existing database for a match, or can be added to enhance the empirical data we have on DNA. DNA analysis on archaeological remains pose some difficulty due to DNA degradation and contamination. An ancient DNA methodology is employed that compensates for this scenario. Sampling process is more arduous, costly, and may not yield enough material for results. So genealogy and archaeochemistry. What can isotopes and DNA do for genealogy? So the relationship between these disciplines are complementary, where both can help fill in information that the other may lack. DNA can provide more concrete evidence of relatedness, provide family tree details, provide unrelatedness, analyze disease and other health conditions, and investigate distant human origins. And when combined with isotopes, they are powerful tools genealogy can use, and vice versa. But what questions can't they help answer? There are limits to any research. We cannot know the past exactly or definitively. We postulate and make informed guesses because we will always be lacking some information. And by adding archaeochemistry data and information, it can only illuminate our knowledge of the past, whether from yesterday or 100 years ago. So let's look at some examples. So applications to genealogy. I'm going to mispronounce this name, the Sviertz-Spork family, were counts in Bohemia from the 17th to 20th centuries of what is the Czech Republic, or Czechia. The modern family wanted to clarify the biological relatedness of a child who died in 1817, named Philip Sviertz-Spork. Due to the degraded remains, the DNA analysis was inconclusive. Carbon and nitrogen isotopes were used to determine dietary similarities between 11 individuals from this family, including 3 young children. Several samples of the same individuals were taken to determine a range of results across various points of their lives. So this was done by sampling multiple points on the teeth. The hypothesis for this type of interpretation is that if these individuals were raised in the same household, their dietary signatures at the same ages should, in theory, be similar. However, Philip's results were dissimilar, while the 2 younger individuals, theorized twins, Barbara and Joseph, were. For the first 4 years of their lives, they had remarkably similar isotope values. However, there was a difference in signature of breastfeeding and weaning. Joseph appears to have weaned around 2 to 4 years of age, with Barbara weaning later at 4 to 6. This could indicate a cultural difference in breastfeeding between the 2 biological sexes. The results suggested that Philip did not have the same earlier life experience as the 2 other individuals, and in fact showed different nitrogen isotopes at particular stages in his life. Ultimately, the results from this analysis contradict some of the death and birth records of these family members. And I think the conclusions are written best, so I'll quote Jan Červík et al. Quote, "The results of the study show that in situations where it is not possible to apply DNA analysis, the creation of an osteobiographical profile and, in particular, the use of stable carbon and nitrogen isotope analysis from childhood has great potential for clarifying biological family relationships. This applies both in bioarchaeology and in the forensic field, and not only to dizygotic twins, but also to situations where DNA isolation is not possible or DNA is contaminated. The present study is also significant to an area of bioarchaeological kinship research and exploring social aspects of kinship, e.g. kin behaviors, beyond the identification of biological relatedness in general, and clarification of genealogical relationships more specifically." So the next example, in 2000, in brushland near Saltaire, Utah, duck hunters found the remains of a cranium, teeth, scattered bone, and a scalp fragment with hair still intact. The remains had decomposed for at least a year, adding challenge to the identification. This was an unprecedented case for isoforensics, as the name suggests, an analytical services company that uses isotope analysis and forensics. This was their first case. Her DNA profile did not initially match any database, and so her identity was unknown. Oxygen isotopes were employed, which are similar to strontium, to determine her movements prior to Saltaire. By using 26 centimeters of her hair, they were able to put together her movements spanning several months before her death. The isotope ratio analysis indicates approximately when she left Utah and when she returned, twice. When she left Utah, she showed values from the Northwest region of the US. And with this guidance, the investigator was able to narrow down where to look for missing person reports. When he found one that fit, further DNA testing coupled with dental records allowed for the positive identification of Nicole McCall, 20, from Tacoma, Washington. An additional individual from Salt Lake City found in a rolled carpet in 1978 was identified by the company, Patricia Barrett from Texas, by using isotope ratio analysis to determine her movements prior to death. Unfortunately, both her and Nicole's murders remains unsolved. Again, one particular issue is being remedied. The tap water in cities may not reflect local geology, and so then the comparative mapping is not reliable. In order to overcome this, isoscapes are created from water and hair samples from metropolitan areas that may provide a more refined map for urban samples. Thomas Kent was born in 1865 in Castlelyons, just outside Fermoy, Cork. On the 1st of May, the Royal Irish Constabulary raided their family home. During their 4-hour-long resistance, a constable was shot dead. David Kent, Thomas's brother, was injured, and Richard, another brother, was also fatally injured. Because an officer was killed, David, Thomas, and William were arrested. They were court-martialed on the charge of armed rebellion. Although William was acquitted, David's death sentence was commuted, and he was to serve 5 years penal servitude. Thomas received the death sentence. On 9th of May, Thomas Kent was executed at the Cork Detention Barracks and buried close to where he fell, in a shallow grave covered in quicklime. He was the only one of 16 to be shot outside of Dublin for their role in Easter week. In June 2015, the National Monument Service exhumed the body, unsure of its identification due to poorly kept records. Traditional DNA techniques were not possible due to the degradation of the remains, and the forced homozygote approach was employed. With the increased sensitivity of this technique, DNA was obtained and then compared to living relatives, and his identity confirmed. On the 18th of September 2015, a state funeral was held for Thomas at St. Nicholas's Church in Castlelyons. There are nuanced calculations when it comes to certain scenarios, like when a population is dependent on aquatic resources and certain other plants like beans that have enriched nitrogen content. These provide challenges when interpreting the results, but with caution and knowledge, this can be mitigated. Evolving technology restricts the possibilities. As with Thomas Kent, this type of analysis on degraded DNA was successful, but only relatively recently has this been possible, and technology continues to advance. There are certain laws that may pose difficulty when looking into genealogy, like Article 1611 of the Civil Code in France that prohibits DNA identification unless for medical or scientific research or in civil cases. However, written consent is always required. In regards to research and funding issues, there can be bias within the researcher. It is their questions influenced by their own interests. And although this provides for passionate researchers, sometimes personal bias can cloud the research. Facility and equipment access significantly can gatekeep research, as the specific equipment required might not be local, and the cost of sample processing can cost thousands, which can then preclude academic research from being conducted. Selective funding based on political or other social affiliation can also undermine research progress due to funding projects with certain outcomes. I know that's actually quite short. So I've touched on archaeology, archaeochemistry, how it can assist genealogy in answering questions about someone's identity. And genealogy can in turn provide a framework for which these approaches can flourish. But to wrap up with a quote that I think exemplifies the human condition, we are all just vessels through which identities pass. We are lent features, gestures, habits, then we hand them on. Nothing is our own. We begin in the world as anagrams of our antecedents.